Good evening, students. Last time we have started with the lesson heredity and variation, and we have finished up to the types of chromosomes. Now you have learnt last time that cell consists of nucleus, and inside the nucleus there are chromosomes, and these chromosomes are visible when there is cell division. So these chromosomes are actually made up of coils. They are coiled and supercoiled and twisted. Okay, so that each of these arms are made up of DNA. You can see the structure of the DNA. So chromosomes are made up of DNA, which are actually containing the information for the synthesis of proteins. And you have learned last time that proteins are responsible for expression of the traits or characters. So therefore we are going to now study about DNA or deoxyribonucleic acid in detail. So let us now study about how was the DNA discovered, who discovered, and who made the model of DNA first. Okay, so and then we will study about the structure of the DNA. Okay, so chromosomes consist of DNA, and this DNA was discovered by the Swiss scientist or biochemist named Frederick Miescher in the year 1869. So he was the first person to discover this acid. Okay, so actually it is a nucleic acid. Okay, and it is present since he found it in the nucleus. Therefore this acid, okay, was called as nucleic acid, since it was first found in the nucleus. It was called as nucleic acid. But actually DNA is present even in other cell organelles also. It was later realized that it is also present in other parts of the cell too, like in the mitochondria also it is present. In the plant cell there is a chloroplast. In the chloroplast also there is presence of DNA. Fine. So DNA is not only present in the nucleus, but it is also present in mitochondria as well as in the chloroplast. But since it was first discovered by Frederick Miescher in the year 1869 in the nucleus, therefore it was called as nucleic acid. So along with deoxyribonucleic acid or DNA, there is another nucleic acid called as RNA. That also we are going to learn in this video today. Fine. Now, so you have come to know that it was first discovered by Frederick Miescher in the year 1869 and in the nucleus. So it was called as nucleic acid. So DNA is actually present in every living organism, whether it is a virus, which is at the age of living and non-living, whether it is not called as living organism, but it is at the age of living and non-living. It is present in viruses as well as bacteria and all other organisms from bacteria. You have learned about the 5 kingdom classification in the case of 8th standard. In 8th standard you have learned, and there are different organisms from starting from bacteria, that is Monera, to the highly evolved, that is human beings. So therefore DNA is present in all the living organisms, including viruses, which are actually not living organisms, but in them also they are present. Why? Because of DNA there occurs cell division and reproduction is taking place, because it's a main function of the DNA. Fine. So DNA is what is the function of the DNA. Okay, so these molecules control the functioning, growth and division, that is reproduction of the cell. So since this DNA is present, it actually divides. It is something called as replication. Because of this process of replication, cell division takes place. And when cell divides, it forms more number of cells, and therefore growth take place. Because the cell division takes place, growth also takes place, and also of the functioning of other cells, other parts of the cell also DNA is very useful. And since it is helping in so many functions, that is functioning, growth and division of the cell, it is also called as master molecule. Remember, it is also called as master molecule. Fine. Now the structure of DNA was first the model or the structure of the DNA was designed by the two scientists, Watson and Crick, in the year 1953. So these were the two people who made this model. You can see how is the model of the DNA. You can see that there is a double, it is like a ladder, but that ladder is flexible, twisted. Correct? It looks like a ladder, and you can see for the ladder there are different rungs are there. So okay, the same way DNA is also, the model of DNA also looks like this. And since there are two strands which are running parallel to each other, but since it is in the form of helix, it is also called as a double helical structure or double helix. Fine. So you can see that there are two parallel threads of nucleotides, okay, are coiled around each other, okay, and therefore the structure is called as double helical structure. So it can be compared with the coiled and flexible ladder. If you just open it, try to open it, it will look like a ladder, but it is, since it is coiled and it is flexible, okay, so it can be compared with a ladder. So I hope so you have understood about the how is outer structure of the DNA. So it is also called as double helical structure. Fine. Now let us see now what are these nucleotides. Okay, so DNA consists of each strand in the molecule of DNA. Each strand means that you can see the rung of the ladder is called as one strand. So each strand, the strand means the two threads of the DNA. Okay, they are called as strands, two strands which are running parallel to each other, but they are coiled. Now each strand in the molecule of DNA is made up of small molecules known as nucleotide, and you can see the structure of a nucleotide. So whatever you can see, those bead-like structures, okay, which are joined to each other, actually they are called as nucleotide sequences, different nucleotide sequence. So one dot means that one nucleotide, okay. So this strand, each of the strand of DNA molecule is made up of small molecules known as nucleotides. Now what are these nucleotides made up of in turn? They are made up of three different parts. They are nitrogenous base, phosphoric acid and pentose sugar. You can see the structure again. You will understand how is the structure. It consists of three different parts, that is, one is nitrogenous base, two phosphoric acid and 3 sugar called as pentose sugar. Fine. Now you can see the structure again here. You can see that there is a phosphate group which is attached to a sugar. So sugar is in between, and at the end there is a nitrogenous base. Now nitrogenous base, why? Because it is made up of mostly the nitrogen atoms. Therefore they are called as nitrogenous bases. Now, in the structure of nucleotide, a molecule of nitrogenous base and phosphoric acid are joined to a molecule of sugar. You can see that there is a pentagonal structure, and therefore that sugar is also called as pentose sugar, which is there in between the phosphate group, and phosphate means PO3. Okay, PO4 is also called as phosphate group, and nitrogenous base in between there is a sugar which is joining both of them. Then these nitrogenous bases now again are of two types. Okay, nitrogenous bases are of two types. One is called as purine, another one is called as pyrimidine. Again, purine and pyrimidines are divided into two different types. Purines are adenine and guanine. You can see the first two structures are called as purines. One is pink in color and the other is in purple color. So that pink color one is called as adenine, and second one that is purple color is guanine, and both of them together are called as purines. Whereas pyrimidines are three types, But in the case of DNA, there are only two, that is cytosine and thymine. There is one more pyrimidine called as uracil, which is present in RNA, not in DNA. In place of thymine, in case of RNA, uracil is present. So thymine is not present in RNA. So you remember, cytosine and thymine in the case of DNA are called as pyrimidines. So again I'll repeat. See, a nucleotide is there. Nucleotide consists of three parts. One is nitrogenous base, second is phosphate group, and third is pentose sugar. Again, these nitrogenous bases are of two types, that is purines and pyrimidines. Purines are again two types, that is adenine and guanine. Okay, you can see the structure of purines. They have one pentose and pentagonal structure and one hexagonal structure. Whereas pyrimidine has only one hexagonal structure, and pyrimidines are again two types, that is cytosine and thymine in the case of DNA and cytosine and uracil in the case of RNA. Fine. So right now we are talking about DNA, so it has cytosine and thymine. Okay. Now, nucleotides are arranged like a chain in the molecule of DNA. You can see how is a chain, you can see that ladder. And these nucleotides are arranged in the form of chain, beads of the chain, as you have seen earlier, as I have told you. Fine. Now the two threads or two strands of the DNA molecule are comparable to the two rails of a ladder. You can see that there are rails of a ladder. Okay, and made up of alternately joined molecules of sugar and phosphoric acid. You can see the third structure which is there. You can see how is the phosphoric acid is joined to the pentose sugar, and pentose sugar is in turn joined to the purines and pyrimidines. Fine. And then each of these rung, okay, each of the ladder is a pair of nitrogenous base joined by hydrogen bonds. Okay, so you can see now over here, you will understand what I am talking about here. Fine. So here you can see that each rung of the ladder, you can see how they are forming the ladder. Okay, so each rung of the ladder is a pair of nitrogenous base joined by hydrogen bonds. You can see that how they are getting joined by hydrogen bonds here. Adenine always pairs with thymine and cytosine always pairs with guanine. You can see C is pairing with G and A is pairing with T, if you can see over here. So that is, they are joining the two strands of the ladder. Two strands of the ladder are joined to each other. How they are joined? It is joined because the nitrogenous bases are facing each other like this. And how these nitrogenous bases are joined to each other? Suppose there is in this strand adenine is present. On this strand, what will come? Thymine will come and join to adenine. Fine. If suppose thymine is there in this side, then what will come? Adenine will come and join. Suppose cytosine is there, then guanine will come and bind with cytosine. Suppose guanine is there here, then cytosine will come and bind. So you have to see how the structure of the DNA is formed when it is getting copied. That is also called as replication. This process is called as replication, when the DNA strand first breaks apart into two, and then since DNA is double helical structure, that means two strands will be always there. Whenever new strand will be formed, you will have to remember that adenine will pair with thymine, cytosine will pair with guanine, and this way second strand will be formed, which is just the complementary of the other. Okay, complementary, just the opposite. Here adenine is there, thymine will be. Here thymine is there, adenine will be there. Cytosine is there, guanine will be there. Guanine is there, cytosine will be there. So like this pairing takes place in the DNA, and the structure of DNA is formed. So this process is also called as replication, which you need not study now because it is at a higher classes or higher level you have to study. Fine. So you just have to remember that adenine pairs with thymine and cytosine pairs with guanine. Right? And when they are joined to each other, they are joined by hydrogen bonds in between the two. This is regarding the pairing, how pairing takes place in the case of DNA. Now let us talk about what is meant by a gene. Now you have learned about a chromosome. Chromosome is made up of DNA. And DNA, as I have told you, is a long strand, because the full chromosome is made up of DNA and some protein molecules. So when you just unwind this chromosome, you will get the DNA strand. And for this DNA, there are small segments. Each of these segment is called as a gene. Okay, each of the segment on DNA is called as gene. So there are small segments present on the DNA, and each of these segment is called as gene, which actually codes for proteins, which actually codes for protein synthesis. Okay, so from here protein synthesis starts. From one gene, proteins will be formed, and that protein, once it is formed, it will be expressing certain character or trait. Now each chromosome is made up of a single DNA molecule, and segment of the DNA molecule are called as genes. Due to variety in the sequence of nucleotides, different kinds of genes are formed. So variety in sequencing of the nucleotides are there, and therefore there will be A, T, G, C, C, A, G, any combination can be there. And since there is a lot of variety in the sequence of the nucleotides, different kinds of genes are formed. So these are arranged in a line. You can see how they are arranged in a line, and genes control the structure and function of the cells and of the body. So they are controlling the structure as well as function of the cells and of the body. Fine. Now also they are helping in the transmission of hereditary characters from the parents to the offspring. So that is called as heredity, as I mentioned to you. You can see the genes are passed on from the parents to the offspring. So that is why certain characters from the parents, like mother, will be shown in the offspring, that is the child, and some characters of the father will be shown in a child. So it is how it is being transferred. It's because genes are getting transferred from these two parents into the offspring, and some of the characters of parents will be shown by the offspring. Okay, hence they are said to be the functional units of heredity. Why the genes are called a functional units of heredity? It's because they are transferring characters of heredity. That is why they are also called as functional units of heredity. Okay, that is why many similarities are seen between parents and their offspring. It's just because the genes are transferred from mother and father to the child. Some from mother, some from father, and it will be a mixture. Sometimes it is a mixture of the two. Fine. So that is why it is also called a functional unit of heredity. Now information about protein synthesis is also stored in the genes. Now you can see how protein synthesis is taking place. Now this you are going to learn when you are studying. You will study about the RNA. What are the different types of RNA? That time you will realize why I have put this picture over here. Here there are three different types of RNAs. Okay, that is, one is messenger RNA, which is that line which you can see, red line is called as a messenger RNA, which carries the message for the synthesis of protein. And how this messenger RNA is formed? Messenger RNA is formed from DNA. Okay, the segment of DNA, that is a gene. From the gene, a single strand of mRNA is formed. That is messenger RNA is formed, and this which carries the message for the synthesis of protein. And other RNAs, there are three different types of RNA. Okay, messenger RNA, that is also shortly it is called as mRNA. Second is ribosomal RNA, which you can see that structure like this dome shaped structure which you can see is also called as ribosomal RNA. And those structures which are T-shaped, inverted T which are coming. So or you can see T-shaped only, they are called as transfer RNA, because they carry, for proteins they require something called as amino acids. Amino acids join to each other to form a long length of protein. Like how for a building block, for building this bricks are there. So like that, for building proteins, amino acids are required, and you can see amino acid, those round structures which are coming, they are actually joining to each other and a protein molecule is formed. So this way proteins are formed by the help of RNA. But this RNA carries a message from where? From the DNA. That is why we say that DNA consists of information for the synthesis of protein. Okay, each of the segment of DNA, that is gene, is transferred into mRNA first, then mRNA is read by ribosomal RNA, that is that structure which is dome shaped, and transfer RNA brings amino acids according to the code what they have. So once they bring this amino acids, okay, they join to each other, these amino acids are getting joined and a protein is formed. Okay, so now let's see about what are RNAs. Now this is one of the fact of DNA, that is DNA fingerprinting. If you have seen CID and all, you might have heard about DNA test and all. So DNA test is nothing but fingerprinting, or the sequence of DNA is done to find out whether the DNA of that particular person is matching with the other. Now the sequence of the genes in the DNA, there are many genes on the DNA, correct na? So the sequence of various genes in the DNA of a person, that is the genome of the person, is identified with the help of DNA fingerprinting. So this is a technique wherein you can find out what type of genes are present in the full length of the DNA. And this is a very useful technique to identify the lineage. Lineage means, suppose the child, there is a child or someone unknown person has been has died, then you can find out from its DNA, from his or her DNA, to whom it belongs to. That means who are the parents of that person. Okay, you can find out by using this DNA fingerprinting technique. So this is also called as to identify the lineage, okay, and identify criminals. Okay, from sometimes when a person dies, okay, the person dies, in that person's fingers, nails and all, the criminal's hair might be there, or maybe the flesh may be there. So from that flesh or from the hair of that criminal, DNA can be extracted and the whole genome can be studied, and the experts, forensic experts, can come to know who is actual criminal from DNA fingerprinting technique. Fine. So it is useful to identify not only the lineage but even to identify criminals, because it is unique to every person. So every person DNA is different. Why? Because it comes from two different parents, and that is why the offspring which is there will be is a mixture of the mother and father. Therefore lineage can be come to know, and each person will be having its unique DNA sequencing. Fine. Now let us learn about the ribonucleic acid. I have almost told you about ribonucleic acid. There are three different types of ribonucleic acid. First of all, you should know that it is second important nucleic acid of the cell. First one is DNA, as I have mentioned to you. Second one is RNA. It is also made up of ribose sugar, okay, that is a pentose sugar, phosphate molecules, and 4 types of nitrogenous bases. Here also same way, sugar is there, phosphate molecule is there, and also nitrogenous bases are there. Now only difference between deoxy and ribonucleic acid is that in the case of deoxyribonucleic acid, there is sugar which is deoxyribose, whereas here it is a ribose. That means oxygen is present here, there oxygen is not present, therefore it is called a deoxy, and this is called as ribose sugar because it has the presence of oxygen. Second, it has phosphate molecule, and third thing, it has nitrogenous bases. Now difference in the nitrogenous bases I mentioned to you earlier also, that this one consists of uracil in place of thymine. DNA consists of adenine, thymine, cytosine and guanine, whereas RNA consists of adenine, guanine, cytosine and uracil. Fine. And DNA is double stranded, whereas RNA is single stranded. So this is another difference between DNA and RNA. Okay. Now the nucleotide here also, it is the smallest unit of the chain of RNA molecule, is formed by combination of ribose sugar, phosphate molecule and one of the nitrogenous bases. Large number of nucleotides are bonded together to form a macromolecule of RNA. So there are many nucleotide sequencing which is forming a single thread, or you can say strand of RNA. Now depending upon the functions of the ribonucleic acid, there are three different types of RNA. So first one is ribosomal RNA. You can see that dome shaped structure which was there. So also called as ribosomal RNA. You can see the structure of ribosomal RNA, how it is twisted. Okay, but it's a single strand. So the molecule of RNA, which is a component of the ribosome, that means ribosomes are present in the cell. If you have studied about the cell structure, you will understand there is something called as ribosomes on the endoplasmic reticulum. So these ribosomes consist of this RNA, and therefore since it is present on the ribosome, it is called as ribosomal RNA. So this organelle is called as ribosomal RNA. Fine. Ribosomes performs the function of protein synthesis, as I have shown you previously one slide wherein the ribosome was reading the message on the messenger RNA, and it was going forward like this. It is because this is helping in the synthesis of protein. Fine. Now second one is messenger RNA. You can see how the message has been read by ribosome. So that ribosome which has joined over here, and you can see amino acids which are joining and forming the the protein which is coming out of the ribosomal RNA. So messenger RNA is that yellow color strand which is read by the ribosome. Fine. So this is protein synthesis actually taking place. But what is mRNA? mRNA is that yellow color strand. Okay. So messenger RNA, the RNA molecule that carries the information of protein synthesis from genes. Okay, from genes, that is DNA chain in the cell nucleus to ribosomes in the cytoplasm, which produces the proteins, is called as messenger RNA. So where is protein synthesis taking place? Protein synthesis taking place in the cytoplasm. But this it starts from where? From the nucleus, okay, to ribosomes in the cytoplasm. So the ribosomes are present only in the cytoplasm, and DNA is present in the nucleus. So in between what is there? From DNA, RNA is formed, that is messenger RNA is formed, and this messenger RNA comes where? In the cytoplasm, and in the cytoplasm there are ribosomes which comes and binds like this and goes on reading the message of the messenger RNA, and slowly transfer RNA brings one amino acid and comes and joins this amino acid in the ribosomal RNA. Okay, therefore we say that ribosomal RNA helps in protein synthesis. Fine. So this type of RNA, which is that yellow color, which carries the message or carries the information of protein synthesis, is called as messenger RNA, since it carries a message for protein synthesis, whereas ribosomes helps in protein synthesis. Fine. Last one is the transfer RNA. As I mentioned to you, transfer RNA is the RNA molecule which, according to the message of mRNA, carries amino acid up to the ribosomes, is called as transfer RNA. So its duty is only to get what? Amino acids. Okay. So it will have a particular sequence for it. Okay, that is also called anticodon, that you don't have to study this year. In the next year you will be studying. So it consists of certain message. 3 different codons will be there. Okay, based on these codons, amino acids will be joined to the transfer RNA, and this transfer RNA along with amino acid will be coming towards the messenger RNA, will be reading the message, and will be joined in the ribosomal RNA, and amino acids will be coming out from the ribosomal RNA and protein will be synthesized. So this is regarding the 3 RNAs and the DNA. I hope so you have understood, though it is complicated, but if you read thoroughly and if you have listen to me carefully, then you will be understanding what I have told you. So next time we will deal with the Mendel's laws or Mendel's principles of heredity. Till then students, bye and thank you.